Hola, soy Jessica Sánchez y esto es Más allá del ADN, un podcast donde exploramos las historias detrás de las ciencias en salud. Aquí conocerás a investigadoras e investigadores que están transformando el conocimiento en impacto real. Hablaremos de su camino, los retos que enfrentan y cómo sus hallazgos pueden mejorar nuestra vida. Porque contar la ciencia es también celebrar a quienes la hacen posible. Welcome to Más allá del ADN. I'm Jessica and this is our first episode in English, so it's something that I'm really excited about. This podcast was born in Colombia with the idea of bringing science closer to people through stories and conversations with those who dedicate their lives to research and education. Today we will take that mission beyond the language, talking about science and the human side of it. So my guest today is Jonathan Soucy. He studied chemical engineering in Rensselaer Polytechnic Institute and did his PhD at Northeastern University in chemical engineering and is currently a postdoctoral research at Madison lab in the Skepenza Research Institute affiliated to Harvard Medical School in Boston. Also, he is an incoming professor at the University of Wisconsin in Madison and he will open his lab soon. Jonathan I met at the Madison lab. And since I joined, he has been incredibly helpful sharing his knowledge with me, which makes this episode more special. So welcome to Más allá del ADN.
Thank you. Excited to be here.
So to start, we can to the beginning. So what brought you to science?
Yeah. So I guess like growing up, so I grew up in Maine. So neither of my parents were scientists, but they both had like pretty good careers. My dad was an accountant, my mom was a nurse, and school was just really valuable and important. And I was always pretty good at math, and then taking like science classes in middle school and high school was really exciting, especially the science fair. I thought the science fair was a really nice opportunity to like be creative. So I actually was building airfoils, so like how airplanes fly, and I was just testing different airfoils to understand which ones would work the best. So kind of a physics problem, a bit far removed from what I'm doing now, but I feel like that's kind of what got me really interested in science. And then going off to college, I was trying to figure out what I all wanted to do, and turns out when I actually took a real physics class, I didn't love it as much as I loved when I was like getting to experiment around with airfoils. But I was also taking chemistry classes, and we made aspirin in our chemistry class by mixing two acids together. And that was like super cool to me because the teacher for my chemistry class was a bit eccentric. So he like licked his finger and like ate some of the aspirin that we made in class, because he said he had a headache. And I'm like, wow, that's super cool. You can make a drug just using chemistry. So that was kind of the genesis of the idea. I wanted to like make drugs to like help people. And so I went to college to study chemical engineering, because that's what chemical engineers do. They make drugs. They do the manufacturing of all of that. They do a lot of other things, but that was the area that I was most interested in. And then I realized that these drugs couldn't work on many diseases because not all diseases can be treatable with drugs. And so I found like the biology side of things, like protein engineering, drug delivery, and other systems like around that. So that's kind of how I started to get into this. I'm sure you have other questions that will be a little bit more specific.
I like to listen, like the story, like how from the beginning, what was like the sparkle, and then you know, that's the idea. So you have worked like across several areas. You mentioned like you started working like in drug delivery systems, and then you started to work with organ in a chips, and then you start to work in neuroscience. Like, how was that? Why did you change?
Yeah, that actually like is a great question that like is from where we kind of stopped previously. But like, so again, it all comes down to like the classes and the teachers and the professors that you meet throughout your schooling, I guess. And I had a teacher at RPI, Rensselaer Polytech, where it was a gene and drug delivery class. And again, it was just like, oh, I want to like learn more about different ways to treat disease, because that was kind of where my interest was. And so gene and drug delivery. And his research was on like neuroscience things. And I'm like, oh, that's super cool. I would love to get into that. So as I was applying to PhD programs, I had originally thought I was just going to do like a drug delivery thing, because that's kind of what I was working on before. I just happened to fall into it because that's what chemical engineers tend to do, drug delivery. But then I'm like, oh, well, neuroscience is super cool. So I want to do something with neuroscience. So in the program that I had gotten into, I had identified a couple of people that were doing neuroscience related research. Through that, I identified Ryan Koppes, who was my PhD advisor. He was brand new. We ended up starting on the same day at Northeastern. But his research was about the neurocardiac axis, so how the brain controls the heart. And so I wasn't super interested in the heart side of things. I was more interested in the neuroscience things, but that was kind of why I was like, wow, this seems like a really fun project that I can really like learn a lot more about neuroscience. And so originally the project that we had come up with together was to build a neurocardiac patch. So the idea behind it is if you have a heart transplant, your nervous system isn't there anymore. Or if you have a heart attack, like the nervous system is all broken. And so it'd be cool if you put a patch on the heart and it would help regrow the neurons to innervate the heart and control it a little bit better. Turns out we really didn't know that much about the cardiac nervous system, enough to build a device that could restore the nervous system in the heart. But so then we took a couple steps back and we're like, okay, so we need to study this system. And I actually read this in like a magazine, like about the organ chip space. Someone had built an organ-on-chip model of the skeletal muscle and then was using the nervous system to innervate it. And then they even went as far as using optogenetics to stimulate the nervous system. So optogenetics is when you can shine light on an excitable cell, in this case a neuron, and then you can control its ability to fire. So you can like stimulate the brain is kind of the idea. So you shine light on these neurons and it stimulates the skeletal muscle, and skeletal muscle with twitch. And it was in an organ chip system. And I'm like, that's super cool. And so I'm like, I would love to do this for the heart. Like just basically the same idea, but put cardiac neurons and cardiac cells and stimulate them with light and just like try to understand how the cardiac nervous system actually functions before we go into this whole I need to build a cardiac patch thing. And so that was like how that project happened. It was like in a magazine and I'm like, oh, let's try that. And then from that area, just switching into vision research. So I was like probably like maybe 2 or 3 years into my PhD. And in the United States, when you are 26, you get kicked off of your parents' health insurance.
Okay.
And so health insurance is, really important. And here in the US, you have to pay for it. But so as a final thing, my parents are like, hey, you should go to all of the doctors and like make sure that like you have all your checkups, because we don't know the next time you'll get to do that. And I'm like, okay, sounds good. Like I was pretty healthy, so I'm like, this should be fine. And so I went to all of these doctors, the physical, the dentist, and then the eye doctor. And so at the eye doctor, so being in Boston, it's a very teaching heavy environment. So it's always like residents that like do the initial exam, and then the attending, the person who's completed all of their training comes in and checks their work. And so the attending, or the resident, was checking my vision, and they were like, wow, you have terrible vision. Like, what's wrong with like your vision? Like, here, like, you need glasses. Why don't you wear glasses? So they were trying to give me glasses. They got a prescription for me. And then the attendant came in, and he was just like, oh, like, huh, what's wrong with, like, you have scarring on your cornea? And I'm like, oh, like, I had, like, an implant when I was younger. And so I was born with a congenital cataract. So cataracts, like, usually older people develop them. But I was born with it. And being from, like, a part of Maine, the doctors didn't necessarily know that children could have cataracts. I didn't even know it was a thing, but, So as a result, it wasn't treated until I was a bit older. So I was about 4 years old when it was treated. So basically they just removed and they put an intraocular lens, like a little piece of plastic in my eye. But because I didn't have vision for the first like 4 years of my life, basically, in that eye, because the cataract was blocking most of the light, I never developed that connectivity like with my brain, between the retina and the brain. It didn't develop properly, and the condition's called deprivation amblyopia. So the first time I heard that term was when I was 25 and like a half years old in grad school, because I had an eye doctor like my whole life, and like they just checked on it. It was fine, nothing huge, big deal. I was a kid, so it didn't mean anything to me, and I'd never heard that term before. And so I was talking to him, and he's like, oh yeah, you don't need glasses because they wouldn't even help you, because even if we correct your vision in that eye, your brain would just still not process it. And so I'm like, oh, that's really cool. So it's basically like my eye innervates my brain and the connection is broken. He's like, kind of, but not really. But yeah. And so I was working on the cardiac nervous system, which is your brain innervating your heart. And so I'm like, well, that would be really cool if I could develop a model system of my own vision to try to understand it a little bit better, try to develop therapies and everything else. And so that's how I decided to pivot my career from the cardiac nervous system, which I was more just trying to get into neuro, to something that I was actually passionate about with the visual system. So basically the connections between the eye and the brain. And yeah, so then I tried to find people that would do that. My advisor, so Ryan knew like a bunch of people in like the cardiac space. He knew some people in the neuro space, but he didn't necessarily know anyone in the vision space. And so I applied to some programs. Through some of these applications, I got to meet really successful ophthalmologists who then actually, so all the way at Stanford, I was talking to some ophthalmologists there, and they made the introduction to my current advisor, Peter Baranov. So he was just down the street from me. I had no idea, but that's how I like met Peter. And then I joined his lab because he saw the benefit of like the engineering approaches, and how I could apply like engineering design principles to the problem that we're working on in the lab, which is retinal ganglion cell replacement therapies for glaucoma and other optic neuropathies. And so moving forward, now I have this tool kit, and I can combine these worlds and build retinal on-chip models. I can continue doing transplants of our retinal ganglion cells. And so that's kind of how I...
yeah. And why did you decide to like stay in research, like do a postdoc, and like what was the motivation behind?
Yeah, so I really wanted to work on this problem. And so when I had finished undergrad, or as I was finishing undergrad, I got an internship at Genentech, which is like the company that I like wanted to work for. It's still an awesome company, and some of my best friends are working at Genentech now. But one of my managers there, he recommended that, oh, you'll eventually hit a glass ceiling, so you should go do a PhD, and then if you want, you can come back, and then you can actually do the exciting stuff here. And I'm like, okay, so that sounds good. That made a lot of sense to me. But then as I worked on my PhD, and I decided that I wanted to study and investigate this visual neurocircuits, I realized there aren't any companies that are actually doing this kind of work, because companies are really profit driven, and so they need to be in order to stay in business. And so a lot of the things that I'm trying to understand are just fundamental basic questions, and not necessarily a treatment for a disease just yet, because we need to understand some of these basic things before we can actually bring it to market where there's like a druggable thing or a tissue engineering approach or something that can actually make it into patients. You need to understand stuff. So it's kind of like the same idea with the cardiac nervous system. I had this idea I was going to make a product, the cardiac patch that helps the neurons grow in, but it turns out, that would have been a great like company idea, but it wasn't a great like project because we didn't know enough about it. And so working on the organ chip space was a way to learn more about the cardiac nervous system. Similarly, with the visual system, we really need a better understanding of like neuroplasticity and how all of these cells are connected and how to grow them back and all of that before we can actually create products. And so there's just not any company that could do that. because who knows how long this is going to take.
Yeah. But that's why it's important, the basic science. Because if we don't have like that research, then we don't have how to develop like the products. So how long have you been in the Barnum lab?
I started in July of 2020. So that was a fun time to start because of the pandemic. So I finished my PhD during 2020. So I got to defend virtually. So that was an experience. But then starting in Peter's lab, we started in July. There was not a lot of interactions because all of the social distancing going on. So we kind of just all like did our own things. I had to like learn to do things a bit more by myself than would otherwise be expected, just because like you couldn't be in the same room as someone else. But I had great people in the lab. There was Julia and Moni, who were other two postdocs in the lab that were there for longer than me. Because of course they were there longer than me. But yeah, so they really helped me, and Peter helped, and so it was a slow ramp up, but it was also really exciting and fun because it was a brand new space. But a lot of what I learned from my PhD was applicable here. Started in July of 2020. So now it's 5 years and a couple months. So a bit of time. So longer than my PhD because my PhD was closer to like 4 years and maybe 9 months. And so now we're...
Wow. And how would you explain to someone that is not in science, like why it's important to link the connection, like, to regenerate the RGCs or like...
Yeah. So... how we see each other today is because of your vision. And so RGCs are the wire that connect your eye to your brain. And if that wire is broken, then you can't see each other. And so we're trying to replace the broken wire.
And how are we trying to do that?
So there's a couple different ways that RGCs can be replaced. The first is stuff that we work on, where we take a new cell and put it in the eye and try to make it grow back and connect. So the idea is basically like just replacing the wire. The other idea is you could take the cells that are already in the eye, change them in some way to convince them to become an RGC, and then they can grow back like all the way. So again, you're replacing the wire, but you're replacing the wire with material that's already there. And then maybe a final way is using bioelectronics, where you could implant a device or maybe strap a camera to your face, but then stick an electrode all the way into your brain and just have it simulate vision. So that would be like a completely artificial, robotic kind of vision. But in theory, that would be another way to kind of skip the wire. But without that wire, your eye can't connect to your brain. But in theory, like if you build like a wireless system, maybe.
Yes. What's one experiment or discovery that made you stop and think, wow, this is actually working? Like, this is impressive.
I guess recently, the coolest thing that I've done that I was like really excited about was I built this mouse model where you can turn off the retinal ganglion cells using a drug. And so I gave these mice this drug, and I was recording the activity from the retinal ganglion cells, and within minutes of giving the drug, like it was completely gone, all the activity from the ganglion cells. So the cells were off. So that was like super cool because it was like such a real-time visualization of what I was trying to do. So that was like the most exciting thing in recent memory. A couple other things were some of the other projects that I worked on in the lab. So like when you transplant, when you put new cells into the eye, you need to get them to go to the right spot in the eye. And so a big part of chemical engineering is using like gradients and transport phenomenon and stuff like that. So what we were able to do is use gradients of chemicals in the eye to control how the cells moved into the eye. So you could put the cells on one side, add a chemical on the other side, and they would be attracted to it. And so it was a way to help position the cells within the retina, which was important. And it was cool that it worked. Because it was like, in theory, it should work, and it was cool when it actually worked.
It's like, oh, it makes sense. Yes, it's working. Yeah. And what would you say, like, are the biggest challenge in restoring the RGCs?
Yeah, so there's a lot of challenges in restoring RGCs. There's actually a whole consortium called Restore, which is devoted to all of these challenges. So the first one is you actually need to make the RGCs. And so you can make them in a couple different ways. You can make them taking stem cells, turning them into retinal ganglion cells directly using just chemicals. You can take stem cells, turn them into organoids, like embryonic bodies, and get them to become RGCs that way. So like mimicking development. You can alter their genes to make them become RGCs, like taking stem cells, just altering their genes, and then they become RGCs. So a bunch of different ways that you can make RGCs. So that's one of the challenges. Another is actually doing the transplant model. So you need the right model for understanding the disease. So we can't test any of these things in humans because there's ethical considerations for that. So you need to rely on either animal models or organ chip models. And having the right model system to test in is really important to test for efficacy eventually for when you need to do it in people. Additionally, so that's the second challenge. The third challenge is getting the cells to survive once you transplant them and getting them to go where they need to go to structurally integrate. So how do you make sure they're surviving and they're going to the right spot in the retina? The fourth challenge is to actually get these cells to connect to the other cells within the retina, and not only just connect to the cells there, but connect to the right cells and the right numbers and the right proportions and all of that. And then the fifth and maybe the biggest challenge here is, well, after they do all of that, then they need to grow all the way back to the brain, and then they need to go to the right spot of the brain and then connect to the right stuff in the brain. And then maybe if they're connected to all of the right stuff, if we go back to the deprivation amblyopia, well, if your cells aren't working for a long time, the stuff in the brain doesn't really recognize it anymore. It doesn't work. So you probably then need to train these new RGCs using some sort of vision rehabilitation. So similarly to how if you break your leg and you need like a rod inserted or something, you need to go to physical therapy so you can learn to walk again. Well, so even if we can get everything to connect and everything to survive and everything to be perfect, we probably need some sort of vision rehabilitation therapy. That, again, we have no idea like what that even entails right now. So a lot of questions, a lot of big challenges. But that's kind of why I think like the organ chip space is really nice for this, because you can perturb individual things in it, and like you can address questions like separately, so you don't have to solve them all at once. And that's why this consortium is really important, because no one scientist is going to be able to achieve all of this. We need a big group of people working on each of these challenges like independently, and then hopefully we can all come together and share our knowledge and actually develop curative therapies for patients.
Of course, the importance of collaboration, because like just one person cannot develop everything. So that's really well. What is the biggest like progress since you started to work in this? Yeah,
so since I started working on this, there's definitely been a lot of progress towards like even developing like human retinal ganglion cells. So when I first started, most of it was like mouse retinal ganglion cells or primary cells. Primary meaning it came from the animal, and so they took cells from the animal and then used them for transplants. There was also like mouse cells, so you take mouse stem cells, turn them into retinal ganglion cells. And so a lot of the progress that has been made is like, hey, now we can actually make human retinal ganglion cells, which is great. And so we can transplant those, and a lot of the progress has been around how to get these cells to like get into the right spot. And so some of the work that we've done with the gradients is some of that progress. Some of the other work in the field that's been done is eliminating barriers. So for instance, some researchers at Johns Hopkins have destroyed, ablated the inner limiting membrane, which is one of the barriers that sits on top of the retina that prevents the retinal ganglion cells from getting in. So if you can remove that barrier, cells get in a little bit better. And so all of that has been like really great. You can also boost the survival of these cells by altering their genes. So that's been another huge benefit in terms of like modifying the retinal microenvironment to improve survival. So a lot of steps have been made in these first three areas, like so making the cells, having really good transplant models to do it in, and then how do we get the cells to survive and structurally integrate. So the big stuff that hasn't really been done is like that connectivity stuff, both in the retina and the brain. There's some work being done in these spaces where you can do endogenous regeneration. So basically the host cells, you can crush the optic nerve, and then by altering the genes, you can make it grow back and maybe restore a little bit of vision. So that's super useful, those people, because now it's like, oh, well, maybe we can do some of those gene modifications in our donor cells, and maybe we can help them grow back. Similarly, if you transect it, maybe you can put like a nerve conduit like in the optic nerve to help them grow back. And so there's ideas that have been played around, and other people are working on them just in different ways. And so I think really combining a lot of expertise would be really cool. And some of the not for RGC replacement per se, but for other diseases in the retina, say like macular degeneration, there's been a lot of progress to transplanting a retinal pigment epithelium to help restore, or even transplanting photoreceptors. And that's like in clinical trials, the transplanting the RPE, the retinal pigment epithelium, and maybe the photoreceptors one is starting pretty soon. But for those, you don't need to have it go all the way back into the brain. You just need to have it connect and go to the right spot in the retina. So maybe a little bit easier there. But it's really cool that this has happened in the past like 5 or so years, where people have been like, oh, I can take stem cells and then I can put them into people. And it's happening in the eye. It's happening about 200 microns away from where we want our cells. So it's pretty close.
Yes.
So that's really exciting that all of that's happening. And similarly, I mentioned like you can do like the bioelectronic stuff. That stuff is happening too. Like people are actually, a couple papers published like even last week. You can implant like an electrode system into the back of the eye, stimulate it with light, and it'll convert that light stimulus into current, and then it can travel up the neurons and restore some sort of vision. Crazy. So like all of this has been happening in the past like couple years. So like the eye is like honestly a hotbed for all of these new technologies because it's so accessible. And what's more important as a sense than vision? So yeah.
Yeah, well, so you have been also mentoring and collaborating. So how do you see the value of communication, like with science and with the public?
Yeah, so all of my research has been funded by taxpayer dollars, like all of it, especially in this vision research community. So it is really important that we like articulate our vision for science very well to the public, because they're the people that are supporting it. I think that publishing like open access is really important because you already paid for it once. You probably don't need to pay for it again just to read the science. So I think that's really important. I think especially like in our times now where like trust in science is at an all-time low, talking about the research that you do in relatable terms is incredibly important. So it's super helpful that I have like a community around me. Even like, so my parents, they're not scientists, but they're educated. They're in their professions like accounting and nursing. And so just talking to them like makes me have to understand, like I can't just talk at like this really high like terminology level. I should use terms that are understandable. And I think like even talking to other scientists and going to conferences, it's better to use terms like that, because then more people can understand. And the point of going to these meetings are like to disseminate your work as widely as you can. So the more people that can understand what you're doing, like the better. And in this collaborative environment that we need to build to do the RGC repopulation work, like it's important that we talk to everybody. Yeah.
Yeah, I agree with that. And I think because for me, sometimes it happens like when I'm talking with my family or with friends, like I feel like they know what I know, but they don't know. So it's like, okay, no, I have to think, how can I explain this in other ways? So yeah, sometimes it's hard because you only know those technical terms, or, you know, like very precise and sometimes it's harder than...
Yeah, and the thing is, we all started out like not knowing any of these terms. So sometimes it's useful to like think back. Like it's like, oh, where was I? Like when I first started here, what would have been useful for me? And I think that's like kind of what helps make like a good mentor. Like a good mentor is like, can you remember like when you were like in that stage, what would have been helpful for you? And yeah, like that sort of communication and like transparency is really valuable.
Yes, that's true. And what advice would you give to international researchers starting like their own journey in the research in the US?
Yeah, so getting into the US system is definitely challenging. I know that because a bunch of people have reached out because I'm starting my new position within the next year or so. So a bunch of people have reached out, and it is really challenging because for instance, even like application fees, like people have different economic backgrounds like that are not in the US, but you're still expected to pay these things. In terms of like breaking into the system, I think it's just a matter of like continuing to like reach out and taking like baby steps to get there. One of the benefits of the pandemic is remote work possibilities are a real thing now. This isn't necessarily like for an international student, but one of my trainees that I've been working with, he is a student at Brown, so not too far away, but all of the work that he does in our lab is all remote stuff. So like I work with him, and like he's a member of the lab, but he gets to work on the data side of things. He gets to write some computer code, he gets to do some image analysis, and it's a way to like have people engaged in the lab without the commitment of being in an expensive city like Boston. I mean, Providence is still not a cheap city, but
but Boston is like...
But say like something like that can be a really helpful thing. Like, hey, can I work remotely for some time? I know that our lab, the Baranov lab, we've had people like, so it's a very international lab, and we've had people that are in like their home countries, and they're still working in the lab because they're able to work remotely. Okay. So those opportunities are potentially really good to like get into the US like science system, I think.
And yeah, now before ending, like congratulations on opening your lab soon.
Thank you.
So that's a huge milestone. And what's the vision behind it? Like it's never describe everything.
Yeah. So my lab, I call it the Nerve Lab. So neural engineering for restoring vision. And our lab will basically use a combination of in vitro and in vivo model tools to study donor-host neural connectivity in the retina, in the brain, and between the retina and the brain, all towards the complete understanding of like neuroplasticity and developing curative therapies for vision loss. So that's the big overarching goal. But yeah, like organ chips, tissue engineering. I like to call it retinal tissue engineering. That's like I feel the catch-all term for the lab. But yeah, really excited to start building this space and creating something new and contributing to science.
So you will still work in vision and in neuroscience, but like what is like that change that you feel like you're doing, like more in vitro, more like...
Yeah, definitely like more on the let's add the in vitro model systems. So one of the things, so we're transplanting human cells into mice, and so I think it'd be really beneficial to transplant human cells into humans, but we can't do that. Yeah. So the next best thing is like, well, can we make a human model system? And so if we could transplant human cells into our human model system, maybe we can use that to probe and optimize for connectivity, like in a dish before moving into people. And so that's like one avenue. And then using technologies like optogenetics and chemogenetics to probe and stimulate, because in the animal models, we have our photoreceptors, so we can just shine light and go through the circuits. But in our in vitro systems, we don't have that. So using optogenetics as like analogs for it could be really powerful. So those are kind of ideas.
And that's very interesting because I feel like nowadays it's like very important to have in vitro models and it's like organ shapes, organoids, not just because of the human model possibility, but also like because we can reduce the use of animals.
Right.
So that's very important.
Yeah, a lot of Europe has like started to move away from using like animal models for like drug testing, and the FDA Modernization Act number 2, which is like an initiative in the U.S. It probably started like maybe 15 years ago, but finally it's starting to actually like happen. The FDA is deciding that, hey, if you can demonstrate your technology in an appropriate humanized model system, you don't need to do the animal testing. And so there will always be a place for animal testing, but I think these are going to be a nice supplement and potentially like incredibly useful for certain diseases.
Yeah, I completely agree with that. And one last thing you would like to share, like if people that are not in science, like you want them to know, like this is a fun fact of science or this is something really interesting, like you need to know this in your life.
A fun fact about science. I guess most of the things that you try as a scientist, it probably won't work. But that's kind of where a lot of fun discoveries can happen. You kind of go down a rabbit hole, like we can go back to my cardiac patch example, where it's like, oh, I tried this thing, it didn't work at all because we don't know enough about this. And then it leads to a whole new branch. So just because it's a failure, it doesn't mean that it's really a failure. It's just a pivot point to something that's probably more exciting. So yeah, that's probably my like tidbit about science. It's just not the published paper you see at the end where it's like scientist discovers XYZ. And then also the fact that most of science now is not one person laboring in a lab all alone. It's a huge collaborative effort. There's so many people that have like worked for this, and that's very evident too by like the Nobel Prizes that have all been given in the past like probably 10 years. They're all to like at least 3 people because there's not a single person that develops anything anymore. Science is very collaborative, and so having open communication with everybody and speaking in terms that are accessible, broadening our scopes to everyone is also like a
And how would you approach like a problem where like you keep failing, like it's not working? What should I do? What should I change?
Yeah, so I guess one piece of advice that I've always received is if you fail, you should fail quickly, because you don't want to like toil away at like one problem forever, because like there's only so much time in the day, there's only so many resources that are available, but if you're failing, like you can, depending on the scope of the thing, you try to pivot to something that is not failing. So, for instance, in grad school, I was doing a lot of electrophysiology recordings, and they weren't always working. And the reason was, I didn't know this at the time, but the reason was the density of the electrodes wasn't high enough. And so when we were seeding our cells, the cells were just not landing on the electrodes. And so made sense. Like I wasn't recording anything because there was nothing on the electrode. But so my pivot from there was, well, the cells should be doing something. And so I tried to like figure out ways to measure them as a different way. So instead of using electrophysiology, which was the plan, I just measured their catecholamine release. So like their release of chemicals. And I was like, oh, let me try to measure them this way. And then, so you can measure them also using electrophysiology. You can use a potentiostat, which looks at like redox reactions and like how things are oxidized. And through that, you can measure the level of these chemicals. You can also like give these chemicals to, so I was also working on heart cells. This had nothing to do with heart cells, this project. But I was like, well, what if I just put this chemical on a heart cell and it beats faster or slower? So it was kind of like a biological sensor. So that was pretty cool. And so probably taking a step back and thinking about another way that you can answer the same question. Because failing isn't, you might fail at the experiment, but the idea is still there. So you can try something different. Similarly, like, so with this mouse model, like that I made recently that I was super excited about, that I could turn off the ganglion cells and turn them back on. It has never worked. Like it's I get one step forward, I'm like, wow, I made it, and now 2 steps back, like, oh, it died. Like, and so I don't know, like trying to figure out like why it died was like just a whole thing. But like once I did, I was able to be like, okay, so the reason why it was not making it is because this drug was interacting with all of the cells in the body and not just the ganglion cells. And I'm like, well, that's unfortunate. And so just by doing like a more targeted delivery approach, I could like localize it. So that was like one thing that was like a one step forward kind of thing. But then it's like, oh, well, it's not working all the time. And so like devising different systems and thinking about it like that. And so just because like it fails, you should fail quickly so you're not like hitting your head against the wall. But then just trying something different to ask the same question, I think is yeah.
Thank you so much. And thank you for sharing your story and for reminding us that science is not just about data, but it's also about collaboration, passion, and a lot of other things, curiosity. Thank you so much for being here.
Yeah, thanks for having me.